Welcome back to the lecture series in animal physiology. So, we are in the 9th week. Today, we will be starting the 4th lecture. So, as of now in this lecture, we have talked about the architecture of the eye and the problems and the diseases encountered at the level of cornea and followed by in the lens. And then we talked about the architecture of the retina and the anatomy of the rod and the cone cells and their position on the retina. And if you look at the previous picture where we talked about the arrangement, lecture, so this is the 4th lecture of week 9. So, if you look at the retinal structure, on the bed of the retinal pigment epithelial cell, you see the rod and cone cells are sitting. These rod and cone cells synapse with the horizontal cells and partly synapse with the amacrine cells. Horizontal cells connect to the amacrine. These are connected to the bipolar. There is a complex network of these cells and layer by layer they go. Then all of them culminate into retinal ganglion cells, and these retinal ganglion cells eventually all their processes travel to the brain, and the spot where only the processes are traveling is called the blind spot, where no image is being formed. So, each one of our eyes has one such blind spot. So, what is interesting to look at is this. So, now on total aspect, if you look at the eye, so if these are the lens and this is the corneal layer, and this is the retinal layer being shown like this. Of course, you have a blind spot somewhere out here. Now, if you look at it, your pigment cells are sitting at the back. So, essentially that means light has to travel all the way to the back to form the image plate, which is very interesting if you think of it. Logically, you will be thinking that it should be present in the front, but actually it is not like that. So, the light has to really travel through, if I give them a different color code, that this layer followed by, say, this layer followed by blue layer. So, these different layers what I have mentioned, like the amacrine cells, ganglion cells, bipolar cells, and likewise none of them has any light sensitive pigment. So, the light is essentially traveling like this. So, in that process a lot of information is being lost, but that the system can compensate for it. It is kind of tuned to compensate for it. So, why is it so? So, if I have to number the layers, if this is your retinal pigment epithelial layer, this is layer 1, this is layer 2, this is layer 3, this is layer 4, this is layer 5, this is layer 6, and of course, this is what is going to the visual cortex of the brain. Okay. So, now when they are sitting on the back, as I am mentioning, in that process they are losing a lot of information. But since none of these cell layer has light sensitive pigments, so they do not respond to the light, but of course they encounter a lot of photic damage or photo damage and oxidative stress. So, this is one pertinent problem which happens. Now, coming back where we left in the last class. So, what kind of processing? So, today we will be figuring out what kind of processing is happening at this layer. That is our key area of the processing which is happening at the rods and cones layer. So, now we will be revisiting the anatomy of the rods and cone cells again. So, this is how we will take the example of rod, but for the cones it remains the same. Okay. So, this is where, now if you look at this anatomy, the places where I am circling, this is where your light sensitive pigments are sitting, which in the case of rods is called rhodopsin, and in the case of cone it is called conopsins. Okay. So, nail down the whole pathway. So, this is how a classic rod looks like. Okay. So, it has a cell membrane, which is the outermost membrane, and then it has a disc membrane. So, what I am drawing now as a oval shaped disc structure, these are called the disc membranes, and on the top of disc membrane are the light sensitive pigments. Okay, something like this, and here you have the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, synaptic terminal. Here is the inner membrane, and here IM stand for the inner membrane. Here you have the outer membrane, right. This we have already talked. Now, if you place an electrode out here, there is something very interesting which happens. If you place an electrode like this and an electrode outside and measure the current across it, or measure the voltage across it, that is a better idea, you will realize that these cells are sitting at around minus 40 millivolt. Unlike most of the cells which sits at minus 80. But this is in a normal condition. Okay. So, there are two situations what we talked about. Okay. One situation is that these cells which are sitting there, these cells goes through two different set of situation. It could be either dark or it could be light, because when the light falls, it is the light situation. When the light is not falling, it is a dark, and within a blink of your eyes, okay, the cell goes through, say for example, close my eyes. So, there is a dark cycle, okay, because there is no light entering into your eyes. So, a rod and a cone has to be. So, let us again get back to this. So, a rod and cone cells responds to light, right. So, your best bet to understand this will be, okay, when the light is falling, it should generate an action potential. Logical, because earlier than this, when we talked about the ear, we talked about that when the sound wave comes, the hair cells bend at a certain angle, and that opens up the voltage mechanosensitive sodium channels, and it works like that. Similarly, in the case of olfaction, we talked about when the olfactory molecule comes and binds on the particular receptor, it opens up certain gated channels. Similarly, same thing is being repeated when you talk about the tongue, when a particular, this is gustatory receptors, when a particular food or particular taste molecule binds, whether it's a sour, whether it's a sweet or whatever, you know, it opens up certain ion channels. By the same token, by the same token, you will anticipate that when the light falls on a rod or a cone cells, it should generate electrical current. So, in order to understand these cells, you have to do an electrical measurements, electrical current, or electrical impulse, and these electrical impulse are should be carried to the next layer, which is the bipolar, horizontal, and likewise so on and so forth. That is what you guess it should work. So, in order to do their electrical measurements, you have to have a system where you can vary the light and the dark. You expose them to a light, you do an electrical measurement. You expose them to the dark, you do an electrical measurement. That's your control. Just like you give a sound wave, you make a measurements from the hair cells. You give a olfactory molecule and you measure from the olfactory cells or olfactory neurons. By the same token, you give a light impulse and you measure the electrical current. Now, it was observed there is something very funny. When light was shined on the rods and the cone cells, very interestingly, it was observed the voltage is around minus 80 millivolt when the light is being shone. So, that means when you are shining light, the membrane is not responding or the cell is not responding. On the contrary, so this is basically breaking your paradigm, right? So, it is not really what is happening. So, there is no electrical current generated when the light is being shined. Instead, when it is a dark, you see the membrane voltage is minus 40 millivolt. It seems that at the dark there is a flow of current, at the light there is no flow of current. Just opposite. So, here be careful, just opposite to the situation what you have come across as of now. Olfactory molecule binds to the nose, it generates an action potential in that olfactory neuron. A sound wave hits into the hair cells of your cochlea or the inner ear. It generates a mechanosensitive activation of the mechanosensitive ion channels, which carries the impulse. So, here just opposite to that, entirely opposite to that, when it is dark, it generates a current. So, let us correct this and then we will explain what does that mean. So, when there is a dark, so this one is not working, right, what we thought. Instead, what is working is when there is a dark, this generates an electrical current and that impulse is being transmitted. Now, what does that mean? So, before I get into that point, what does that mean? For the first time when this was discovered, it was given a term called dark current. So, it means the current is generated in the dark. Now, having said this, I come to a different aspect to trying to explain it. We always say when an impulse comes, the electrical signal has to be generated. But the thing is that when we see an object, we see a contrast in an object. Say for example, what that means. So, if you look at the geometry of the eye, say for example, image has to form out here. What we will be seeing, so the image is forming out here, right. So, image is forming out here, some form of image is forming. So, the image which is forming, say for example, so the lights are traveling something like this. Here the light is reaching faster, here the light is reaching later than that, here the light is reaching even later than that, here the light is reaching even later than that. So, likewise what you are seeing, that there is a contrast which is being created in the whole image, whereas out this spot you will have a. So, say for example, this is the object, this is the shape of the object, okay, and this is what you are imaging, and this is where your plate or recognition plate is sitting. It could be a CMOS, it could be a retinal layer. So, the light will travel, think of it, the first one, the first set of lights will come like this, these are the first one based on the topography of the structure. These three will kind of, or maybe, so followed by the light will be coming. So, if these are the first rays which are coming, followed by something like this, these will be the later ones, these will be even later ones, and maybe some of these will be coming, right. Followed by this, there will be some stuff which will be coming much later than that. So, in that process what is happening? If you try to imagine this, you are seeing a three-dimensional structure in a contrasting fashion. So, there is a contrast which is being created. So, if these are the first set of lights which are reaching here, 1 1 1, and if these are the second ones are, say, these ones, 2 2 2, third one is 3 3, like it. So, I mean there could be millions of such points which will be hitting out here. So, what will happen on this stuff, there will be an image which will be forming, like, you know, first of all the data points will be formed here, 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 and these information will be traveling, well, followed by the very minor micro nanosecond gap or picofemtosecond gap, the second set of signals will be coming, second set, then comes the third set of information which will be. So, in that process what will happen, that in this vicinity, these places will remain dark, there will be current flowing from in this, whereas this place will become blank. So, say for example, now try to visualize this. This is something called an understanding what you have to do. Say for example, these are the sensors sitting. There are two ways I can make a figure out of it, and let me make the figure. Okay. So, one way is this. Right. So, you are seeing a something like this. You are seeing a number 7. So, in that case all these are, I am darkening it out. I can do the reverse thing, right. What I can do, the reverse will be exactly the same figure. Let me put it side by side. I can make another 7, but in a different way. And this is all the understanding you have to gather. This is something you have to visualize, how I can make it. So, I know these ones I am not darkening, okay. Just let me rub this one, okay. And let me now darken the other ones, okay. Okay, okay. You realize what happened? So, here also I am getting a 7. So, there are two ways you can get a 7 here. As a matter of fact, for anything you can get, either you make the foreground bright or you make the background bright. Is it making sense? try to visualize it. This needs a visualization. So, in this case, what happens? Now, based on that, are you making the foreground bright or you are making the background bright? Let me give you an example of this. If you make the foreground bright, then this is a situation where this is the case, where your foreground is all bright, like look at this one. Or if you make the background dark, so now this is the situation where your background is dark. So now imagine these dark cells are photoreceptor cells which are electrically generating dark currents and sending a signal at a particular point of time. Whereas these 1, 2, 3, 4, 5, 6, 7, these 7 are not sending any signal, right? Not sending any signal also means a signal. Why? Because just look at it. By not sending signal, also it is generating a pattern. So this is how you have to imagine how on the photoreceptor surface the cells which do not, mark my word, do not generate action potentials or do not generate a current when the light falls on them. But remember, on their background there are photoreceptors which are continuously generating the dark current. And so based on the background current, background cells, it generates the image on the surface of the photoreceptor, and that's what makes it so very special. Having said this, in the light of this conversation, in the next class I will take over how the computation happens. Okay, so I'll close in here. In the next class we'll follow up here, because you needed to think over this thing, because this, once this concept is clear, it's a very simple concept, then everything will make sense to you. Thank you.